Sometimes I feel out of place living here in Utah. People pick me off as having an accent also. I did grow up in northern Minnesota and regions of North Dakota, where one of the things I did a lot of was travel around playing youth hockey. I still enjoy playing and coaching hockey here in Utah, and one of my favorite things is challenges is trying to keep up with the Aggie hockey players in their off season. I get to do that. But since I'm not really very good at that, I have to have a real job, and I work as a research geneticist at a lab here on campus at Utah State University. I feel a little bit out of place in this conference also. It's going to be a much different talk than what many of the other things I've heard here. But I think you'll find that I think many of you will recognize some of the plant materials that I will talk about, and I hope you'll find that interesting. And I'd also point out that a lot of the breeding and genetic approaches that I'll be talking about, especially toward the second half or latter part of my talk, this is the same kind of work that's being done and funded very, very heavily from DOE on species like switchgrass, miscanthus, even tree. I'm less familiar with trees, but I know there's a lot of DOEs funding similar genomics breeding work in hybrid poplars, some of the cultivated tree species. I'm not that familiar with it, but do recognize that a lot of these breeding and genetic principles that I'll talk about today certainly are being used in other species, in biofuel development funded by DOE. All of my work is funded by in-house funding through the Agricultural Research Service. We have not had any DOE funding. The title of my talk was given to me, and it's fairly appropriate, except I would point out that our lab is a federal lab. We have a regional assignment. We do testing of plant species throughout many different parts of the western United States. It's certainly not limited to Utah, and although some of the species that I'll talk about are native to Utah, they're certainly not limited to Utah. The Forage and Range Research Laboratory has about 30 employees, including 12 scientists, and we're organized into 4 working groups. One of these is targeted on this biomass and biofuels. It's not limited to biofuels. It's more generally biomass. I'll talk about that in a minute. We have another group, rangeland ecology and restoration, and we develop the mission of our laboratory is to develop plant materials for the West. We have a logo and a brand here, Plants for the West. We develop plant materials for turf grass, low maintenance turf grass, especially emphasis on low water use for urban landscapes in the West, irrigated pasture plants, native plant materials for rangeland revegetation, and then this group is really targeted towards developing plant materials for maximum productivity and forage quality on dry lands, low input plant materials. So the vision of this biomass working group is identify genes, traits, and species for development of new varieties or novel hybrids to improve low input biomass production or forage quality in the western U.S. Some of the problem areas that we work on is the productivity and persistence of legumes in rangeland plant mixtures. We're developing new varieties of tall-statured forage kochia and grasses for fall and winter forage. We work on adaptation and forage quality of plants grown on phytotoxic soils. These are it's for mineland reclamation, developing plants that won't uptake too much arsenic or cadmium or copper. Places like the Clark's Fork River Basin of western Montana. There's selenium contamination on soils in southeastern Montana or southeast Idaho. We're trying to develop plant materials that can grow on those sites and not present toxic forages for wildlife or livestock. And then one of the last things we're working on is this low input perennial grasses for biomass or dedicated biomass or biofuel crops in the West. So one of the problems as we see it is that switchgrass and miscanthus have been heavily promoted as the universal biomass crop for marginal lands throughout the U.S. I've seen websites where they show, adaptation zones of switchgrass all the way across the United States. Well, the western United States is widely dominated by cool season grasses with special adaptations to salinity, drought, and other harsh growing conditions. Most of the precipitation in our region comes during cold winter and spring or fall months. We have the heat units to grow warm season plants, but we don't have the water during that period to grow warm season grasses. So switchgrass is, contrary to what people sometimes say on the news or whatever, popular media, switchgrass is not native to this region and has not been adequately tested throughout much of the western United States. The large Basin wildrye, Leymus cinereus, is the largest native grass in western North America. And I'm sure some of you probably recognize this grass. It's a very long-lived, tall bunch grass that grows up to 3 meters tall in some environments. It once dominated the fertile regions where soil and water accumulate, like floodplains of the Humboldt River in Nevada, or even here in Cache Valley. This is a seed production field we have in Cache, right here in Cache Valley. It probably was a dominant grass in Cache Valley when the settlers came in. The pioneer stories talk about grasses that tickled the bellies of the horses and stuff like this. This was probably one of those. This was certainly one of those grasses, and as you can see, it can grow to quite impressive stature on our valley soils here in Cache Valley. It grows well. It's a lot of these areas where it once grew are now cultivated farmlands, just like the tall grass prairies of the Midwest. These were the best growing sites for plants, and now they're largely gone, but you still find this plant on alluvial soils of mountain foothills and valleys, and sometimes you'll see it, pretty nice swards of it, even in very dry deserts of the Great Basin. But it won't really, it won't form a real productive stand. It'll just be a plant here and a plant there. Sometimes you'll wonder, well, how can a plant that big grow on some of those really harsh sites? But there's going to be a part of the season there in that environment that, where that land is flooded, or maybe there's subsoil moisture that you can't see that can sustain that grass. Some DNA fingerprinting work done by a USU graduate student, May Cullenber, in my lab, identified three different races of Basin wildrye using our germplasm collections from western United States. And there's two different ploidy levels, a tetraploid and an octoploid. The octoploids tend to be larger plants. They're more in the Pacific Northwest region. And the Great Basin ecotype is strictly a tetraploid. Creeping wildrye is a closely related species. It's relatively short but strongly rhizomatous grass, well adapted to saline meadows. It's one of the most salt-tolerant grass species in the world. Actually, all of the Leymus wildryes are very salt-tolerant, but this species, really, that's what it likes to grow in these saline meadows. It was once thought to be a dominant species in the Central Valley of California, where it's still cultivated as a saline biomass crop. Some problems with it. It has recalcitrant seed germination. It has strong seed dormancy, and it's also a poor seed producer. But it does recover well because of the rhizomes. It recovers well following grazing, cutting, or other disturbances. DNA analysis of 600 DNA samples from that collection of plant materials of Western United States shows that there is hybridization between. So each one of these bars, this is a bar chart, each bar shows the ancestry coefficient of 600 different plants, and some of the plants have a mixture of ecotypes. This is creeping wildrye. The blue, red, the red, green, and yellow are the ecotypes of Basin wildrye. There is some evidence of hybridization between the species in some collections. Two of these are artificial hybrids, and then there's also some hybrids that we find in nature. So there is hybridization between the ecotypes and species. And we find that hybridization to occur primarily in this region of southeast Oregon, northwest California, northern Nevada, and parts of Idaho, where we find all of these different species and ecotypes growing together. We find some hybridization. Artificial hybrids of creeping and basin wildrye show a heterotic combination of tall plant height. Here's the hybrids. They show a heterotic combination of tall plant height from basin wildrye and a profusion or proliferation of rhizomes and tillers from creeping wildrye. So this is one creeping wildrye plant that's spread out here from rhizomes. And the hybrids show a combination of those two traits. I see Ryan here in the audience. So miscanthus is one of he talked earlier here at USU, gave a seminar. I'm talking about miscanthus, which is one of the heavily promoted biomass crops. It's also a hybrid of a rhizomatous species and a cespitose species like that. So that's kind of a paradigm that's been shown to be productive in biomass crops, making hybrids of cespitose and rhizomatous species. We evaluated the biomass of these creeping wildrye hybrids by comparing them to 37 accessions representing the parents, creeping wildrye and basin wildrye, as well as 8 other reference species, including switchgrass, which we thought might be productive in this region. We did the plots were 5 plants per plot with half-meter spacing. They were established from transplants. We tested them in 2 locations, one in Teutonia, Idaho, and one here in Cache Valley, with replicated plots. Important point I want to make is we evaluated for 5 years, 4 harvest years, 1 establishment year, 4 harvest years, no irrigation, no fertilizer in any of these experiments. I'm reporting the value. The values here are shown in megagrams per hectare. If you like to think in terms of tons per acre, it'd be divide those numbers by 2. In the first evaluation year, the hybrids, the very best species was tall wheatgrass, and then intermediate wheatgrass and reed canary grass. Reed canary grass you'll recognize as one of the tall grasses that grows along our canals. So basin wildrye and creeping wildrye, first evaluation year, are certainly not our best, but they were better than switchgrass. In the second evaluation year, the native wildryes start to catch up with these introduced grasses. And in the third evaluation year, the single best entry in the study was the hybrids. Basin wildrye also did very good. Here's creeping wildrye. In the fourth evaluation year, again, the hybrids and basin wildrye were the best entries by far. And these biomass, it's not great. It's not what you'd get growing miscanthus in Illinois or something, but at 6 tons, at roughly 6 tons per acre, you're approaching something that is at the level of what they think they need in a dedicated biomass crop from grasses. Those were averages by species. If we look at this by variety, we had some different varieties of basin wildrye in the study and different varieties of creeping wildrye. The hybrid that I made and that I used in those studies was a hybrid of this variety crossed with this variety. And so we had some heterosis in that hybrid. Now that kind of beckons the question, what would happen if we cross this better creeping wildrye with this better basin wildrye? This would be, if we got the same level of heterosis that we got here, this would be the projected yield of a hybrid with better varieties, if we use better hybrid varieties as parents. So in fact, we did that. We made those crosses in, I think, 2009, got them established, and in our first harvest year, that hybrid did, in fact, pretty much perform the way we predicted. Here's basin wildrye varieties. Here's my old hybrid here, and that new hybrid there. So it did give us the yields that we were kind of expecting. So what this basically shows is that I think there's quite a bit of opportunity to breed grasses for improved biomass in our region. The studies I showed you were all based on transplants of clones or plants put on half-meter centers, in part because I didn't have seed of the hybrids. But we've been doing studies in parallel here, developing methods to produce hybrid seed. And that's what you see here. This is 3 rows of creeping wildrye, and these are grown from one plant. And it's actually, this goes on for about a quarter mile. I say it's the world's largest grass plant. This is all one plant here. It's because it's so rhizomatous, we can really clone it easily. In the greenhouse, we just tear that plant apart, make all these plugs, and we transplant it into the seed field. And because it's outcrossing, because it has to cross with something else, we plant the Basin wildrye variety next to it, and then the only thing it can cross with is the Basin wildrye. So we get a uniform F1 hybrid seed population from that. Kind of like corn hybrid production, which I worked for a corn company when I first got out of graduate school, so I kind of carried that idea into this. As I said, one of the other objectives, one of our main objectives of our group is identify genes and traits controlling biomass heterosis. Our hypothesis is that the tall plant height, large stems, and large leaves of Basin wildrye combined with the prolific rhizome production of creeping wildrye is what produces the biomass heterosis. So to identify the genes and traits controlling these complex traits like biomass, we have to try to simplify, develop a simple experimental system. Mendel's first law is that every individual, whether it's a plant or a human or whatever, possesses a pair of alleles for each trait, and these alleles are randomly transmitted to the progeny. One of the alleles is randomly transmitted to progeny. So our hybrids, for any given gene, get one allele from triticoides. I use kind of like Mendel did, or like you did in high school. I use a T for the triticoides allele and a C for the cinereus allele. The hybrid for any given gene gets one triticoides allele, one cinereus allele. We took the hybrids and back crossed to triticoides to make what I call the TTC families. Those individuals either have a combination of TC or TT alleles. We crossed hybrids to cinereus to make other experimental families, either have a TC or a CC allele. Try to simplify the system. Okay, so Mendel's second law is the genes for different traits are transmitted independently. So it's not exactly true. If genes are on the same chromosome, they're not independent. But if they're on different chromosomes, they transmit independently. Leymus is a polyploid with 2 sets of 7 chromosome pairs. So there's 28 chromosomes. It passes it can pass either the cinereus chromosome on to its progeny or a triticoides allele. So for this chromosome, there's going to be recombination between chromosomes, and it'll pass one copy of it onto the progeny. To detect and map genes known as quantitative trait loci or QTLs controlling biomass, what we do is we make genetic maps with DNA markers, thousands of DNA markers to cover all of the chromosomes. And then we also, so each of the progeny, we genotype the progeny with DNA markers to make a genetic map. And we measure the phenotype of those progeny in replicated field trials. So those field experiments I talked about earlier, we also had 300 plants from our experimental families in those field trials. We developed DNA markers by sequencing genes, expressed gene sequences from the rhizomes of the hybrids. And we sequenced 15,000 genes and made several thousand PCR markers. We made genetic maps with 14 linkage groups and over 800 markers that form 14 linkage groups. So that's a map of the 14 chromosomes that we get from the DNA markers. We can align those markers to species like barley, and we number our chromosomes, the linkage groups. Leymus chromosome 1 corresponds to barley chromosome 1, Leymus 2, barley chromosome 2, etc. We can align them to 360 markers to the genome sequence of Brachypodium. Brachypodium is the DOE model grass for biofuels, or one of them. So with those genetic maps, what we can do, so this is basically a map of DNA markers across here for the 14 linkage groups, and we're scanning across the genome for effects on plant height, rhizomes. So the height of that curve is, if that red line goes above this dotted line, we think there's a significant gene effect, say here for plant height, significant effect for rhizomes on chromosome 3, significant flowering genes on chromosome 4 and 6, biomass genes here on that align. So here we see a biomass gene that aligns to the plant height gene, a major plant height gene. Here's some biomass genes that align to our rhizome genes. So what we're doing here is we're comparing the location of genes controlling biomass and other traits. We also look at the progeny for variation in composition, fiber, cellulose, hemicellulose, and lignin. We see significant variation among species. And we can look at genes controlling the synthesis of cellulose, hemicellulose, and lignin in our Basin wildrye or our hybrid progeny. And one of the interesting things we found is that the single of the 800 genes that I had in our genetic map, the single best marker for fiber, that is cellulose, and lignin was the caffeic methyltransferase gene, lignin biosynthesis gene, which is the same gene that's mutated in the corn and sorghum brown midrib mutants, as well as it's a gene that's been used for transgenic downregulation of lignin biosynthesis in switchgrass, tall fescue, in other species. So, you know, what I'm trying to say is a lot of the genes that are being identified in these other biomass crops, we have evidence here that we can use those genes for breeding and modification of yield or compositional traits in our native grasses. So in summary, the best introduced species, intermediate and tall wheatgrass, showed superior establishment in biomass potential, at least in the first 2 harvest years, but the native Basin wildrye and creeping by Basin wildrye hybrids showed superior persistence in biomass production, at least under low input management in the 3rd and 4th years. The hybrids provide a simple and efficient system to identify genes and traits controlling biomass heterosis, as well as composition and other functionally important traits in perennial grasses. I think it's a good model system for biomass crops like miscanthus. I know that unlike miscanthus, our hybrids are fertile, so we can do these experimental. We can make these experimental populations. The miscanthus hybrids are sterile, so they can't do some of the things that I talked about. And then lastly, I'll point out that there's ample evidence of genetic variation that can be used to breed new cultivars and hybrids, as well as engineer, do genetic engineering to lower lignin composition or something like that. So with that, I thank you for your attention.